Human DNA Isolation at Home. These are the necessary materials to make the practice. To obtain my DNA, I find myself here rinsing my mouth. The next step is putting dishwashing soap so it can destroy the cell membranes of the living tissue that we are using. It dissolves fats or lipids, which is the main component of the plasma and nuclear membrane of cells. To continue with the practice, here I find myself putting distilled water and salt. Now I am putting pineapple juice in the same glass where I put my DNA, so the acidic pH of the juice and the enzyme that the juice contains, that is called bromelain, can attack and degrade the rest of proteins and cellular components, which allows us to separate them from the material we are looking for, that is my DNA. Now I am putting the mix of distilled water with salt in the same glass with my DNA and also the mix of the dishwashing soap. As my final step, I am adding alcohol very carefully and slowly through the walls of the glass to avoid mixing and get too well differentiated layers, since it will be in this separation between the mixing layer and the alcohol layer where the DNA precipitates. Since DNA is soluble in water, but when it is in alcohol it precipitates, for this reason, my DNA strands begin to become visible at the interface between the mixture and the alcohol.